Welcome to the lesson on buffered solutions. Buffers are an important part of chemistry, and especially when we talk about physiological chemistry, meaning chemistry in your body or in an animal's body or in any living organism, because an organism cannot survive if the pH changes drastically. And so we have to have chemical ways that our bodies can manage different changes in pH, and buffers are the answer. So let's take a look. So why are buffers important? So if you look at any cell, if you change the pH of the solution it's in, the chemicals inside will not only stop working, but sometimes they will even break apart or denature in a different pH. And so we don't want to destroy our different chemicals in our body, our proteins that are in our body, or fish or anything like that. And so it's really essential for us to be able to take care of these changes in pH that we might experience. Your blood actually has to be at an average of 7.4, and so it's like just tiny bit on the basic side, and we will actually do a whole lab on blood buffers this Friday. And if you talk about your stomach, now your stomach is very acidic, and that's between 1.6 and 1.8, because without it, the acidity, you can't actually break down everything you eat. And also, very interestingly enough, one of the reasons that you do not get sick from food, even though it could harbor dangerous bacteria, is that as you eat, your stomach kills most pathogens that could potentially be in your food. And so if you've ever eaten something, that you probably shouldn't have eaten, but you didn't get sick, you can probably thank your stomach acid for the fact that it killed off anything bad that was left in that food that might have harmed you. How does your stomach handle such a low pH? It actually creates a lot of mucus, and so the lining of your stomach is covered with mucus, and that keeps the acid from being able to react with the actual tissues in your stomach, because the stomach itself would burn if you got acid next to it. What is it called when your stomach burns a hole in it? Well, sometimes if you're really stressed out or you have certain conditions, you won't make enough mucus, and the acid can actually burn the lining of your stomach, and that's called an ulcer. And so that's why ulcers can be dangerous. And if you have an ulcer, you can't eat acidic foods anymore, because that just makes the situation even worse. And so it's an important piece of your life to have buffers in your body. All right, so what is a buffer? So these are solutions that they resist changes in pH if I add some acid or base. Okay, so it doesn't mean that these are strong. It just means that in the buffer you have a chemical that can react with acid and a chemical that can react with base, so that no matter what you add to the solution, it can be neutralized. And so that's why it's a mixture of a weak acid and its conjugate base, or a weak base and its conjugate acid. When choosing a buffer, the key is that it has to be at equilibrium, meaning that there has to be some of the weak acid and there has to be some of its salt or its conjugate base. And so if that weak acid and its conjugate base, or a weak base and its conjugate acid, if they are there, they're in equilibrium. And the key is that there's some of each, which means that the weak acid can accept any hydroxide added to the solution. The weak base can accept any hydrogen added to solution, which means at the end it doesn't change the pH when acid or base is added. That means that we have to be able to pick something that is not a strong acid. So which of the following is not a buffer system? Well, here's acetic acid, that's in vinegar, that's a weak acid and its conjugate. This is also a weak acid and its conjugate, and hydrocyanic acid, that's a pretty strong one. So that is not a buffer system, because it will prefer this side, and so if all of the HCNs are already dissociated, then it cannot behave as a buffer. All right, so did you notice that I said it's important that we have equilibrium? Well, that's true. The mixture of ions in a buffer are the thing that allows it to resist changes in pH, because there's a little bit of everything present in the solution. And its capacity to resist changes in pH is called the buffer capacity. And what that means is there's only so much you can add. If you take a buffer solution and you just pile in a whole bunch of hydrochloric acid, it will eventually become acidic. It cannot absorb every single hydrogen ion if you put a ton in there, but it can absorb to a certain amount. And so in your body, you're not going to accidentally, get a huge amount of hydrochloric acid in your bloodstream. But you may have a medicine that has a byproduct or a metabolic product that is acidic or basic, and your body needs to be able to manage that change in pH, and it's able to do that through this buffering system. At the bottom of your page, I will add this link so that you can take a look. You can pause right now and watch it, or you can wait and watch it at the end. All right, so if you take a look at this buffer system right here, this actually should be at equilibrium, so we should have a double arrow. And what you see is that you have, over here, you have the conjugate base. And so since the conjugate base is here, it could accept hydrogen ions. And here we have the acid, and so it can accept hydroxide ions. And so if I use this up, we will get a left shift, and we'll make more of it to replace it. If I use this up, we'll get a right shift, and we'll make more of this to replace it. So as long as we haven't exceeded the buffer capacity, we can keep adding hydroxides and hydrogen ions to the solution and find that our pH doesn't change. Now I want you to think about it then. If I had a beaker with the buffer, what would be present in that beaker? Here's my beaker. What would be present in the beaker? Let's take a look. Well, we know that our reaction has 4 different chemicals in it. So we know we have our water, our hydronium ion, our acetate ion, and our acetic acid. But we have to have many of each one in order for this to be a successful solution of a buffer. And so you'll see that over and over these repeat. And the wonderful thing is, because we have such a big mix of reactants and products, if an acid is added, this weak base will accept those hydrogen ions. And if a base is added, the H3O can neutralize it, or the acetic acid can also neutralize a base. So these are the reasons why a buffer is so wonderful, because it can act in multiple ways in order to neutralize no matter what is added. So if you look at the blood, you have carbonic acid, which is a weak acid, its conjugate base, which is the bicarbonate ion, and a hydrogen ion. And so as you add OH or H+, you get these reacting back and forth in order to create a perfect solution. So I will also put this link at the end of this lecture. So we are at the end, so let's do a quick learning check. Which of these statements doesn't describe a buffer? All of them describe a buffer except ... Go ahead and pause while you figure it out. There's one of these that is definitely not the answer. So first of all, a solution that resists changes to pH is exactly what the definition of a buffer is. So that must be true. A solution that has a pH approximately equal to the pKa of the weak acid. Now, this is a new term. We didn't really talk about pKa's often, but this is true, and we will talk about that later. A solution containing almost equal amounts of a strong acid and its conjugate base. Wait a minute, a strong acid? That can't be right. We don't work with strong acids, so this must be the incorrect answer. And here, it looks like we didn't exactly finish writing out this question. Solution containing almost equal amounts of a weak acid and its, let's say, conjugate base. And that is also true. So we've had a good day. Go ahead and watch the 2 additional videos that are linked at the bottom of this lecture, and we will see you next class. Thanks so much.